let's talk about the immune system. This is probably my favorite system, so I can't just spend one video on it. This is part 1. There will be a part 2. Watch the part 2 also. So anyway, we split the immune system into innate and adaptive immune systems, and the innate immune system is what you are born with when you come into this world, the immunity you already have. It's also non-specific by definition. The adaptive immune system is going to be specific stuff that you develop as you are exposed to more pathogens. So the innate immune system includes both physical barriers, chemicals, and cells, so not both, all. But anyways, so physical barriers are going to be things like the skin, or like the mucous membranes in the reproductive, urinary, respiratory, gastrointestinal tracts. There's going to be mucus secreted by those epithelial cells, and that's going to protect against pathogens. There are also chemical barriers, like in your saliva and in your tears, there's something called lysozyme, and that's going to break down bacterial cell walls. Or in the stomach, you have a really low pH, so pH around 3 to 5. Not many organisms can survive that, so that's really important for our immunity. We also have some cells that go on in the innate immune system, and one important class of cells, they're called phagocytic cells, and phago means to eat, so these cells are literally going to eat pathogens like bacteria and viruses. And the way that they do this is they're going to send out these little protrusions, and these little protrusions are going to be facilitated by the cytoskeleton. So if you've learned about like actin polymerization, that's what's doing this. And they're going to send out these protrusions and basically surround the pathogen and bring it in so that it's in a vesicle. And then that vesicle is going to fuse with a lysosome. And the lysosome, in case you forget, is a digestive organelle with a bunch of hydrolytic enzymes that are going to break down this bacterium until it's just a bunch of chemicals. And I found this cool, very aesthetic picture of a macrophage, and it's eating a bacterium. And I thought it was interesting because look how big that bacterium is in comparison to this macrophage. I mean, the macrophage is bigger, but the bacteria is pretty darn big. And this macrophage is able to send out these protrusions and completely engulf the bacterium and destroy it. And I just think that's so awesome. So, there are some other cells that are involved. When you are exposed to bacteria, that tissue is often going to undergo something called the inflammatory response. And the reason why it's inflammatory is because the blood vessels in that area are going to dilate, and that's what makes it look all red and gross. And the vessels dilate for a reason, because we want a bunch of immune cells to migrate to this site so that these bacteria can go bye-bye, right? So we do have some phagocytic cells in our tissues. They're called macrophages. So macrophages are going to be in pretty much every tissue. There are also some other cells in the tissue called mast cells, and mast cells are going to secrete this chemical called histamine, and histamine is going to get these vessels to dilate. And when these vessels dilate, other cells are going to come through the bloodstream. So neutrophils are phagocytic cells that circulate in the blood. They're part of innate immunity, and they're going to come in through this process called extravasation, which basically means they're going to leave the blood vessels. So there's actually this really cool whole protein mechanism that they use to get through the endothelium, which is the cells that line the blood vessels. So there are these proteins called selectins that are on the endothelium that these neutrophils can bind to. And then once they bind to them, they can detach and reattach and detach and reattach, and they go through a series of steps, and they get to the other side, and they are extravasated. And they go in and they eat up the bacteria, and then your wound or your inflammation will heal and things will pretty much go back to normal. So we've been introduced to some of the cells, but I want to give kind of an overview of all of the cells that are involved. And all these cells that are involved come from this process called hematopoiesis. And hematopoiesis is the differentiation of hematopoietic stem cells. So a stem cell is a cell that has potential to become lots of different things through a process called differentiation, where basically its chemistry changes, and that changes the genes that get expressed. So you start out with this precursor, and it can make anything. It can make an innate immune cell, it can make an adaptive immune cell. I have this little box that's that's the adaptive immunity. So these cells called B cells and T cells, we're going to talk about them in just a minute. This precursor cell here can become those. It can become those macrophages that we talked about. The macrophages have got this precursor called a monocyte. This cell can become a neutrophil, which circulates through the blood. It can become a dendritic cell, which is a different kind of phagocytic cell that participates in innate and adaptive immunity a little bit. It's not an adaptive immune cell, but it like helps along the adaptive immune system, and dendritic cells are going to be like in the lymph nodes. There's also these things called natural killer cells, and natural killer cells are going to help us with our viral infections. So if a cell is infected with a virus, it's actually going to present proteins from the virus on the outside of the cell. And there's a specific response to it too, but what natural killer cells do is they go, okay, this protein is from some virus. I don't know which virus, but I know it's a virus. So this cell has got to die. So this natural killer cell is going to secrete chemicals that are going to cause this cell to undergo apoptosis, which is programmed cell death. There's also eosinophils, and eosinophils work with multicellular parasites, so they're going to secrete a bunch of enzymes that kill the parasites. So if you've got like a worm or something inside of you, that's where the eosinophils come in. And then there's this small branch of the hematopoietic stem cell line that is not immune cells, it's just components of the blood. So if you have this chemical called erythropoietin, then it's going to differentiate this stem cell into a red blood cell, or into platelets, which are these little like fragments of cells that float around in the blood and help with clotting. So that's basically all the cells that are involved in these processes, but I want to go more in depth into the adaptive immunity, which is what these B cells and T cells do. And this is going to be very specific. So it's going to be different cells responding to each different pathogen that you encounter. And once you have an adaptive immune response once, then the next time you get the pathogen, it's going to be so much easier for your body to respond because you've already set up the specific adaptive immunity for that pathogen. So let's take a look. So like I said, there's two cells involved in this. They're called B cells and T cells. The reason why they're called B cells and T cells is because the B cells mature in the bone marrow and the T cells mature in the thymus, which is like this little immune system organ in your chest near your heart. But anyway, these both have very specific receptors. So these receptors are going to have conserved regions, which basically means that they're going to be the same throughout all the cells. So this here is the conserved region in the B, and this is the conserved region in the T cell receptor. And then they've got variable regions, which are up here. And the variable regions are where things are going to bind. So both of these receptors are going to end up binding antigens. And the variable regions are variable because every single B cell and every single T cell has a different combination of amino acids at the ends of their receptor in these variable regions, and that different combination of amino acids can bind a different antigen. And an antigen is just a piece of a pathogen or a piece of something that is infecting us that can be recognized. And an epitope is the little piece of an antigen that actually binds to stuff like these receptors. There's also of note is that the B cell receptor, which is a little bit more celebrated in the immune system. When you learn about it, you tend to focus more on the B cell receptor. There are heavy chains, which are these guys in the middle, are heavy chains, and then there are light chains. So you should know about the heavy chains versus the light chains. And then you should know that the antigens are binding here and there. And for the B cells, there are actually soluble or not membrane-bound versions of these receptors, and they are called antibodies. And that's something that you've probably heard of before. Antibodies are going to go out in the bloodstream and throughout the body and neutralize pathogens by binding onto them so that the pathogens can't really bind to your cells or do stuff with your cells. So there's two branches of adaptive immunity. One is called humoral immunity, and one is called cell-mediated immunity. And humoral immunity is going to be the job of the B cells, and it's called humoral. It's not the humerus like the bone. It's because people used to think that we were made of like these different fluids called humors. They would call our fluids humors. It's very medieval sounding, I know, but it's because these B cells work in the blood, and they like work directly on the pathogens. Cell-mediated immunity has to do with our own cells that are infected, and that's what a certain class of T cells called cytotoxic T cells is going to deal with. And we're about to learn how these things get activated and what exactly they do. So this all starts with a cell called a helper T cell. So there's two types of T cells, cytotoxic that I mentioned before, and helper, which we're going to look at now. So a helper T cell gets activated by what we call a professional antigen presenting cell. So a professional antigen presenting cell is a cell that its main job is to eat up pathogens. So we talked about phagocytosis. These cells are going to do phagocytosis and spit out a little part of them called an antigen on a molecule called an MHC2 complex. So that stands for major histocompatibility complex. So these professional antigen presenting cells, B cells are actually one of them, but we're going to table B cells for now. The other ones are dendritic cells and macrophages. And dendritic cells and macrophages are going to eat up pathogens, so they're going to kill them, and then they're going to present these things on MHC2. And the reason that professional antigen presenting cells have MHC2 is because the rest of your body has MHC1s. And we're going to talk about why that is. It has to do with killing our own infected cells. But I like to think that the cells that present antigens, and that's all that they do for a living for our bodies, they have the upgraded version. They have MHC2. Whereas all the cells in our body that are generic and do other things most of the time unless they're infected, they have MHC1. So anyways, these MHC2 complexes on the dendritic cell or on the macrophage are going to present out an antigen, the little bit of our pathogen, and the helper T cell with its T cell receptor, so we looked at the T cell receptor up here, it's going to bind the antigen right there. And it's got this little accessory protein called CD4, and CD4 is going to help out the helper T cell in binding this antigen. So we've got the T cell receptor, we've got CD4, and those two things are binding to the MHC2 complex containing the antigen. And once that happens, it's going to send this whole signaling cascade through the helper T cell, and the helper T cell is going to say, I am now active, and the active helper T cell can go and activate other things. So it's going to activate the B cells, and it's going to activate the cytotoxic T cells. So the way that a helper T cell activates a B cell, we talked about this part. MHC2 complex presents an antigen on a professional antigen presenting cell, aka a dendritic cell or a macrophage, and it binds to helper T cell, which has T cell receptor and CD4. T cell gets activated, and that helper T cell is going to now look for a B cell. And the B cell has to undergo this other process first. So before it can bind to a helper T cell, a B cell has to encounter this pathogen. And when it binds this pathogen, it is going to engulf its receptor and the pathogen. So the B cell is going to swallow up this whole thing and get it inside its cell. And once it does that, it's going to do something very similar to what this cell did. It's going to take a little piece of the pathogen, called an antigen, and it's going to present it on an MHC2 complex. So now this helper T cell can come along with its T cell receptor and its CD4, and it can bind this MHC2 complex presenting an antigen. And if the B cell has encountered the pathogen and bound the pathogen, and if it has presented this antigen, and a helper T cell has come and found it, if all these things happen, the B cell can be activated. And when the B cell is activated, it kind of splits into two categories. So it's going to divide and grow and divide and grow, and it's going to make memory B cells, and the memory B cells are going to sit there and wait for the next time you get infected. And it's going to make plasma cells, and plasma cells are going to go out and secrete the soluble or not membrane-bound versions of these antibodies. So these are the plasma cells, and they're going to secrete these antibodies, and these antibodies are going to bind to the pathogen, and these pathogens, anything that's infecting you, it's infecting you by interacting with your cells via its structure and its proteins, right? So if these antibodies come along and block its structure and its proteins, then all of a sudden these pathogens can't really do anything to your cells because they're too busy binding to these antibodies. So that's how the B cells work. Helper T cells, if we remember, they also activate these guys, which are called cytotoxic T cells. Cyto, cell, toxic, toxic. So they're going to be toxic to your cells, or more specifically, your infected cells. So remember back when I said that every cell in our body, actually every cell in our body that has a nucleus in it, so like not red blood cells, is going to present this complex called MHC1. So they're not professional, they don't get the upgraded 2, they get just the basic MHC1 complex. And when a cell gets infected, it's going to say, I need to take one for the team, I need to die so everybody else can be safe, and I'm going to present this antigen on my MHC1 complex. And cytotoxic T cell is going to come along with its T cell receptor and with this other protein called CD8. So remember, helper T cells had CD4, these cytotoxic T cells have CD8. And it's going to bind, and it's going to signal this cytotoxic T cell to start releasing things that are going to kill the infected cell. So it's going to release this thing called granzyme, which is going to start to like break this cell membrane, and it's going to release some other stuff called perforins, and it's going to signal apoptosis, which is programmed cell death. And so this cell has died. And that's how cell-mediated immunity works. So remember that the B cells were part of humoral immunity, or immunity in your blood, and these cytotoxic T cells are part of cell-mediated immunity. So this is basically a review of everything that we've just gone over. So we've got humoral immunity and cell-mediated immunity, and so you're going to have an antigen, and it's going to be taken up by an antigen-presenting cell. This antigen-presenting cell is going to activate a helper T cell, and it's also going to activate the cytotoxic T cell. The helper T cell is going to go and activate these other things. It's going to activate here, and it's going to activate here. And the helper T cells are going to, they're going to reactivate themselves and make more memory T cells. And all these responses are going to combine in order to neutralize the pathogens and in order to kill your infected cells. Now before we end, I want to talk really briefly about where all of this is going on at. So you have blood circulation, right? Here's the heart, here's the arteries, here's some capillaries. I'm not a good artist, okay? I'm sorry. And here's the veins, right? So these things are pretty closed off, but what they do is they let out a lot of water. So there's really high pressure in the beginning of your blood, and it's going to let out water into your tissues. And so eventually there's going to be a really high amount of water in what we call your interstitium, or the tissues outside of the blood. And what that's going to do is it's going to make an imbalance. So we need a way to take the water and put it back in the blood, or else the blood is going to be too low in pressure and you're not going to be able to get out oxygen and nutrients and all these things to your cells anymore, because blood pressure, adequate blood pressure is essential to what we call perfusion, which is like getting out oxygen to all your cells. So the way that we fix this is something called the lymphatic system, and the lymphatic system basically it has some ends that are closed off. So this isn't like, this would be like an end by a bunch of cells. So if these are your cells, you would have a lymphatic thing, a lymphatic vessel that is going to come out this way, and it's going to connect to something called a lymph node. And a lymph node is just going to be a little ball of lymph, which is this fluid that comes from the interstitium. And in here is going to be your immune cells. So you have like some dendritic cells, you have some macrophages, and some B cells, and some T cells. And all of these things are going to be exposed to pathogens, because one important thing to note is that the lymph vessels are more permeable than the blood vessels. So if you've got like a bacterium floating around in the interstitium, it can get into the lymph. So let's say that here's a little bacterium. It's going to float into the lymph, and it's going to end up passing through a lymph node. And then this lymph node is probably going to get swallowed by a macrophage, which is going to spit out an antigen, and that antigen is going to get detected by the B and T cells, and it's going to activate an immune response. So that bacteria, basically at this stage of the game, it goes bye-bye because it gets swallowed by one of these phagocytic cells. And this lymph goes on, pathogen-free, and it's going to eventually connect in the circulation really near your heart. And there's a bit of membrane here that is not completely permeable, so just the fluid goes through and it's going to go back to your blood. But the lymph nodes is where a lot of this stuff is going on, at least when we start this adaptive immune response. In the next video, I want to talk about what happens before we get here, and how the B and T cells actually arise with so much diversity, and how they get to be advanced and mature enough to live in these lymph nodes and kill these bacteria. So I'll see you in part 2.